The overall goal of this procedure is to prepare sex-stratified hippocampal primary dissociated neuron cultures. The first step of the procedure is to sex and dissect embryos on embryonic day 18 to obtain sex-stratified primary neuronal cultures. The next step is to prepare dissociated neurons from the neuronal tissue. The final step is to plate the neurons at the appropriate density and grow them for experimentation. Ultimately, this method, in conjunction with other protocols, can be used for any study in which sex is thought to be an important determinant of outcome.
This method can help answer key questions in the gender research field, such as whether male and female rodents respond differently to neuronal ischemia.
Of the several required preparations, there are a few worth reviewing, such as the tissue culture plates preparation. Dilute poly-D-lysine to 0.1 milligrams per milliliter. Then, under a laminar flow tissue culture hood, generously cover the bottom of each well of a multi-well culture-treated sterile tissue culture plate with the diluted poly-D-lysine. Incubate these plates overnight at 37 degrees Celsius. Second, be sure to wipe down the dissection area, the counter, and scope with 70% ethanol. Third, fill a large flat dish with ice. Label, and fill enough 60 mm dishes with cold dissection solution to keep the male